does that protein function differently? Is it regulated differently? Right? So you can generate mutations. You can also use site-directed mutagenesis to introduce an epitope, right, for affinity chromatography that we talked about. Right? So we can add a 6-his tag using site-directed mutagenesis. Or say there's a domain that you suspect is a kinase domain, right? What happens if you delete that kinase domain? You can use site-directed mutagenesis for that, right?
It's more so like investigative rather than the other method was like to produce insulin for our like the informed for
Yeah, so here you can use this for investigations, right? So you can investigate the function of different residues or different regions of the protein. But right, that can lead to other outcomes, right? So say that you're trying to improve insulin, right? So here, this proinsulin, this is just a gene that we have. Now, the reason why, insulin doesn't cost $1 is because the gene that is being used to synthesize, modern insulin is not simply proinsulin, it's been modified many times, right? Using methods like site-directed mutagenesis to either have longer-lasting effects or to be stable at different temperatures, right? So these genes can be modified to produce outcomes that have pharmaceutical relevance, let's say. Or we can use them in the lab to examine, regulation of certain areas of a protein or anything like that. So site-directed mutagenesis is used to create proteins with new functions or different functions, right? So it can be used for any number of things. All right, so now on to DNA sequencing, and we'll end here, right? So DNA sequencing, yes. I do want to talk about Sanger sequencing because that's the most relevant. Next generation sequencing is kind of interesting. If there's time for it, we'll get to it. Next Friday, we're doing cards, which usually doesn't take very long. So I'll see if I can cover some more of this on Friday. But I do want to talk about DNA sequencing. Now, traditionally, the way that DNA sequencing is done was developed in the 1970s. It's very old technology, but it's good enough. Okay, it does very well. And the way that Sanger sequencing works is by using these dideoxynucleotides. Okay, what is dideoxynucleotide? It's a 2', 3' dideoxynucleotide. So it's missing the 2' hydroxyl like all deoxynucleotides are. But the dideoxy is also missing the 3' hydroxyl. And if you remember, the mechanism for DNA synthesis from last week is the 3' hydroxyl that must be present at 3' end to attack the alpha phosphoryl group of the mu base, releasing pyrophosphate and forming the phosphodiester bond, elongating this piece of DNA, right? So what's going to happen then if there is no 3' OH, as there isn't in a 2', 3' dideoxynucleotide like we have here? What's going to happen? Is DNA elongation possible? No, right? There's no 3' OH to do the attacking. So how are we using this to sequence then? Well, we're using this at the same time as we're using regular deoxynucleotides, 2' deoxynucleotides. Right, so say that we have this sequence here, ATCA-CA-GTG. We don't know that sequence, but we know that that's the region of DNA that we want to sequence. And we know a region of DNA that's upstream of that sequence, okay? So what do? Right? Well, we just do a PCR, essentially. What we do is a PCR, so we denature these by heating them up, right? Now, we add this sequencing primer. And we only need one sequencing primer as opposed to 2 PCR primers because the point here is not to amplify, but instead to sequence. So we only need one sequencing primer. And that sequencing primer should be complementary to the sequence that's upstream of what we're trying to sequence. Okay, so what's going to happen here? We'll add our sequencing primer. We'll add a mixture of dideoxynucleotides and deoxynucleotides, with the deoxynucleotides far in excess. So what we'll get is mostly normal elongation because we have mostly just regular 2' deoxynucleotides, but every once in a while, randomly, a 2', 3' dideoxynucleotide is going to be incorporated. And when that dideoxynucleotide is incorporated, that terminates the synthesis of that particular strand, right? Because there's no 3' hydroxyl that can be added onto. It's 2', 3' deoxy, right? Now, where that happens is random. right? Just depends on frequency, statistics. So what we'll get in the end is a population of DNA strands that all end at different places, okay? We can separate them out by size. And we end is something like this, right? We have one that made it only one nucleotide before a dideoxynucleotide was added. We'll have one that made it 2 nucleotides away from the primer before a dideoxynucleotide was added, one that made it 3, one that made it 4, and so on and so on. By tagging each of the 4 dideoxynucleotides that we added with a different colored fluorescent marker, each of these strands will fluoresce in a different color, right? So if it ends in a T, it'll fluoresce pink. If it ends in an A, it'll fluoresce green. If it ends in a G, it'll fluoresce orange. And if it ends in a C, it'll fluoresce blue, right? So now we can take all these many, hundreds of different DNA strands, all of which were terminated at different points randomly, and separate them out by size, and we end with something like this, right? So we can use electrophoresis to do this. Up here in our well, right, we'll put our mixture of DNA fragments. The longer ones will be towards the top, and the smaller ones will be towards the bottom, right? Each of these fragments will be fluorescing in a different color, depending on which fragment it was, or which nucleotide rather it was that terminated the synthesis of that strand. So let's say that the last base here was a C on the small fragment, right? Well, if this ends in a C, that means that this entire fragment should be blue, right? If this ended in a G, this entire fragment should be orange. If this ended in an A, this should be green. And if this fragment ended in a T, this should be pink, right? So what can we do? Well, we can actually measure the level of fluorescence in all 4 colors as we move from the bottom of our gel to the top. And because the smaller fragments are running toward the bottom and the larger fragments are at the top, we're reading from small to big. So we put this together and say B, blue, orange, green, pink. And if we know the correspondence of the dideoxynucleotide to its fluorescent color, we can say, what is this? Blue is C, orange is G, green is A, and pink is T, right? So by measuring the color now, we know the sequence is CGAT at the beginning, right? So we can do this. This would be running towards the bottom, this would be running right above it, this would be running towards the top, so we can scan from bottom to top and say, all right, well, this is pink, green, orange, pink, right? We can say, well, this sequence is TAG, right? So this is how Sanger sequencing works, and this is how most of DNA sequencing is done. Because it's cheap, it's fast, it takes about a couple hours, and there are many facilities, even here in New Jersey, that do it. You know where that Target is in Piscataway? Right behind it is a company that does this. A lot of Rutgers companies use during COVID. Actually, they allowed us to just drive over there and drop off the samples because they weren't sending out delivery guys. Now we can just drop it and pick it up. Yeah.
Why would the level of fluorescence intensity be different between the same base, like the 2 T's in the middle, if they're different heights? Is there a reason?
The 2 T's where?
They're like pink.
Like these ones?
Why would they be different?
Well, the intensity is the intensity, right? So this varies from fragment to fragment. It just depends how many fragments you have.
Oh, okay.
So how many fragments you have, again, it's going to be kind of random, right? There should be kind of an average baseline, but here you just happen to have a little bit more of this fragment, which is over here, right? So if this fragment just happened to have 5% more or whatever, in this fragment we had about 10% less, let's say, right? So that's just a random variation. But there's a clear peak, right? Where here we only have pink and nothing else, right? So no matter how many of that particular fragment we had, that's the base of that position, all right? All right, so we'll finish up with next gen sequencing if we can on Friday, and I'll let you all go because I can't cover it in 30 seconds. So, yes.
Do you have office hours this week?
I have office hours every week.
Yes, we have Wednesday.
Monday and Wednesday.
Sorry.
Yes, yeah. It is bronzy.